Okay, our next speaker is Maria Leptin. Maria was at LMB from 1984 to 88, first as a postdoc and then staff scientist. And in 1989, she became a group leader at the Max Planck Institute for Developmental Biology in Tübingen. And since 1994, professor at University of Cologne. And from 2010-21, Maria was the director of ENBO, and now she's the president of the ERC.
Pleasure to be here again. I'm, of course, also, like my wonderful colleagues here, often asked for career advice. And all I can say is don't ask me, because whatever I planned during my life never happened. And I always did something different. So when I was still in high school, I wanted to be an interior decorator. Then I wanted to be a ballet dancer, which I actually started professional training, but then switched to training to become a high school teacher and got a degree in maths and biology. When the time came to go into serious teacher training, I freaked out and decided I couldn't possibly go to school every day. And so instead did a PhD at Basel Institute of Immunology, which was a fantastic institute, but it was a very, very tough experience because my idea of science was you go to the lab, you do an experiment, you think deeply about the results next day or maybe next week, and then come up with the next great experiment. I hadn't figured out that experiments sometimes don't work. So it was a very, very tough experience, tough 5 years, even though I had a wonderful advisor, Fritz Melchers. And at the end of it, I wasn't sure I wanted to stay in science. But the Basel Institute at the time paid their PhD students, of which there were only 3. It was 50 scientists, 3 students. So you didn't exactly have good company for talking you through difficult periods. They paid to go and interview for postdocs, and everybody had been saying postdoc is just the best time of your life. So I thought, well, I'll interview. And I went to go to Chambon, for instance, and I didn't go there because he said the group meetings were Saturday morning at 8. Is that true? No way. Friday night at 10, anytime, Saturday morning at 8, no way. So, and I came to the LMB to interview with Peter Lawrence, but Peter's lab was full at the time. He said, there's this guy down the hall, Mike Wilcox. He's working on monoclonal antibodies. That's what you're good at, isn't it? Go and talk to him. Last thing I wanted to do is anything to do with monoclonal antibodies ever again. But because this was the way to come here and see the LMB, I said, well, okay, I'll have a look at it. And I talked to others. I went to Schaffner and to Pete Borst. I mean, it was a time of transcription regulation. But guess what? I talked to Mike, and here he is. I talked to Mike Wilcox, unfortunately died much too young. And I loved the work. I loved the lab. And so that's where I went. Now, somebody said, Joan, I think you said you got a bench this big. There wasn't a space that big in that lab. So what they did, it had sort of struts underneath. They put a board underneath. That was my space. Later, we moved to another lab down the hall, which was much bigger, really nice. Rob Kay was next door. It had the disadvantage that the incubators were out in the hallway. And Max Brutz, who can't eat unripe bananas, would ripen his bananas in those incubators. So we had a constant stream of rotten bananas in the lab. Anyway, so Mike had discovered cell surface antigens that were defined through monoclonal antibodies that defined compartment borders in the Drosophila wing disc. So Peter Lawrence had, of course, worked on compartments. The way the insect body was divided up through genes was a big deal at the time. And so Mike had found a marker for these. And my project was to figure out what these cell surface proteins were. They were only known through antibodies, so it required a lot of biochemistry. And I purified them. I did a lot of 2D gel analysis, looked at their biochemical behavior, purified them, and sequenced them. And this was actually an interesting paper because you see Rudy Ebersold on there. Rudy Ebersold had also been a graduate student in Basel. And I'd met him there, and he by this time moved to Lee Hood's lab, and he was developing protein sequencing of filters, off glass filters, onto which you could transfer from SES gels. And so we tried it on our proteins, and it worked. And this was actually the first time this was published. And you see he's on here without the hood. I mean, things worked differently at the time. He did it in Lee's lab. He was independent. Anyway, so we had the N-terminal sequence, which is completely illegible here, but that allowed me to try and clone it, which involved a lot of techniques and a lot of sequencing, which you may remember we did by hand at the time. And in fact, the sequencing machine where your gels would come out took the place of what otherwise is the water cooler or the coffee machine nowadays, for many conversations and some jokes. I never managed to clone the genes. That was later done by another graduate student in the lab, Thierry Bogard, which is a bit crazy because if you look at the first few amino acids, it actually has my name in it. There you go. So I also met a really important friend and colleague here, Ruth Lehman, who was a highly experienced fly geneticist, had been recruited to a group at the Whitehead Institute straight off her PhD at Tübingen, and taught me proper Drosophila embryology and genetics, maternal effect genetics. And together with her, I then figured out the genetics of what these integrins do in the embryo, and showed that they are actually responsible for cell adhesion and for sticking embryonic germ layers together. So we were serious. We had wonderful times together, really talked science all the time, as I did with Cynthia Kenyon, by the way, with whom I had a private journal club, just she and I, and we'd either go to the orchard and do our papers there, or we'd go to the University Arms and have tea and do our papers there, including the paper by Ed Lewis on homeotic complexes, which is unintelligible, but we managed. But very serious science, but of course, lots of fun all the time. So Ruth is here in one of her sillier moments, and this is one of the parties we had with, you see lots of other female who became Hilary Nelson, Peggy Goodall, who became international group leaders. So before I go on with my science and talk about the last bit of science that I've done on early morphogenesis, I'm just going to show a few pics. I think that's more fun. And we hear that. We've heard about the scariness of the second floor. I mean, I was scared too. And of course, one of the most scary things were the Wednesday seminars. Oh my God, you know, the divisional seminars, everybody had to present, no exceptions. And they really made you quake in your boots. And here's some photos from them where you see the group leaders at the time. Here's Cynthia, Mike Shen, Jim Priest, Peter is there, etc., etc. I think, however, on this case, we were actually not in a scary seminar, but we were celebrating once again. I don't know what, it's too early for the Nobel Prize of these two guys, but so once again, serious science and lots of partying. Partying, the biggest party we had was the 25th anniversary party. What you see here on the left is a banner that we hung down the stairwell that Peter Sorger and I made in the halls of the building. We'll see it here some more. We actually made two. I was going to bring them. I still have them. I should have given them to the LMB. I'll send them. And hung up. And this was the 25th anniversary party. It was not the 20th anniversary party. The party was very posh at a college in town. Jim Watson gave a speech in which he praised himself and complained that the city of, he told us a story that he'd gone to the place where he lived and a young woman came out and he said, "Do you know who lived here?" And she said, "No." And he was shocked. And he said, "Do you know who I am?" Oh, he was even more shocked. And he was even more shocked that Cambridge had not seen fit to put a blue plaque for him on the building. So we talked at this dinner, which was, I don't know why I was invited, almost none of the junior people, and certainly none of the non-scientific staff were invited. It was just all posh stuff. So Peter and I decided this won't do, and we had a party for organized a party for the entire LMB, and here's a bunch of pictures. You see Ron Lasky there. I mean, it was just fun. The champagne was drunk out of buckets. I'll leave it for a moment for you to see. Here's a whole bunch of others. And the organizing team, apart from Peter and myself, had also Tony Hyman. You wouldn't believe this, Tony Hyman, up there on the left as well. And we financed it through these T-shirts and sweatshirts that we sold that Sandra and David were wearing yesterday, of which I also still have a bunch in my drawer, and which I, by the way, designed. Okay, LMB wouldn't be anywhere without the wonderful support staff. Here's a bunch of them from that time. Here's the hallways in which one hung out and had conversations with Barbara. Here's the TMV we heard about earlier. Here's the hemoglobin, of which there's also a copy downstairs, and canteen, you know, and now just, you know, a bunch of people, all these wonderful people who used to be there, Kim, Mike, John visiting, wondering what Mark's up to, et cetera, et cetera. Okay, we're nearly back to science, and here we are. So, the genes that, the proteins, the integrins that had become identified and that I'd helped in figuring out their function at the end of my time here, I had thought they would be involved in a very different kind of tissue morphogenesis, namely in creating germ layers and allowing them, and that this was because of the complementarity of their expression. So I became interested in how the germ layers are made, and this is how it happens. This is a very old slide. An early embryo, which in cross-section you can see is simply an is my cursor visible there? Yes. So I don't have to be patient with this thing. Patience is not my strong suit. This is an early Drosophila embryo, 6,000 cells, very simple epithelium, which over the course of only 20, 30 minutes folds and creates this bubble inside, which then spreads out and becomes the mesodermal germ layer. So when I started my own group at the Max Planck Institute in Tübingen in 1989, I found out that this happens by a series of cell shape changes, stereotypic cell shape changes. And during the ensuing years, my lab, and as well as that of Eric Wieshaus in Princeton, we identified, okay, what's expressed here is a transcription factor called TWIST that actually directs this process. And what we did then was to figure out what are the target genes that the transcription factor controls. We figured them out. And it turns out what these genes do is two things. They relocalize, you see here the adherence junctions in pink. They relocalize the adherence junctions to the apical part of these cells that change shape. And they also relocalize myosin, which you see here at the base of these cells, again to the apical side. And that leads to the constriction. And the genes are all expressed throughout the mesoderm. So the inner cells fold, and we'll hear more about this. They're expressed here. And what Adam Martin in Eric Wieshaus's lab had shown at the time is that as a result of these gene activities, the apical end, so the outer cell, part of the cell, constructs this contractile pulsatile network that leads to the constriction of the cell surface. Here's the word autonomous. We had shown that it's autonomous in the sense that only these cells do this, and the rest of the body is passive. However, even if they're passive, then the rest of the body has to sort of participate. And that's what I did in the last bit of my career on this. I did many other things in between, is to figure out how does the rest participate. And I think I won't have time to go through it all. We did this through very sophisticated 4D imaging set up by Matteo Rauzzi, who managed to image cell outlines throughout early development. And the way we did this was then to sort of peel off the surface of the embryo to be able to look at it. And you see here the tissue folding happening, but you can also now see what the other cells of the body are. They're flowing towards it, they're changing shape and changing size. And in fact, if you can now measure everything, you can measure speed, you can measure size, you can measure anisotropy, and you can see here size shown from small. Everything starts in the middle, green. Some cells get small and some get big. And if you look at this fold, then you see that the ones in the middle that make the fold, it's now shown the other way around, get small, the others stay put. But then you notice that the ones next to this fold get very, very stretched. They get very long. In fact, the ones that are closest to the fold get stretched strongest. But the mesoderm actually goes from here to there. So all these cells will do the same thing in the end. They have the same instructions, but they do different things now. I'll skip through the quantification. So why is that? Adam Martin had measured the levels of myosin in these cells and actually had shown that they accumulate sooner and to higher levels in the middle and nothing out here at all. Well, there you go. That's the explanation then. These cells out here don't have myosin, so they can't constrict. But we looked at this more carefully, actually now looking not from the belly side, but slightly from the side where we can see these lateral cells much better. And it, and it turns out, okay, not running. Well, I don't have time anyway. So we looked at this more carefully, and I'll just show you the quantification now, and looked at these cells row by row, early, how do I do this, late, and quantified initially size. So you see at about just after 200 seconds in this movie, the inner cells start constricting and the outer cells start expanding. Now, if we look at their myosin over time, we don't see the very inner rows here. We just see the outer rows up to row 9. You see that they, in fact, a little bit later, all accumulate myosin. So now that's a problem because now we see that all these cells have myosin, and yet this one constricts, and the one which has just a tiny bit less expands vastly. So how does this happen? Again, all these cells have all the gene projects that they need. Is it perhaps the levels? So here's again the sizes. If you see, the levels in the inner ones do go up earliest, but the outer ones actually accumulate levels that are very high as the inner ones do when they constrict. It's also not concentration. It's also not the functionality. These networks are pulsatile. Well, maybe these aren't. Maybe they can't pull, but they are pulsatile. And you see here a pulse, and you see how it pulls on the membrane. So they're active. They have actual, proper active myosin. So what is it then? We actually then decided, intuitively, we can come up with ideas why this might happen, but we decided to look at it in a proper viscoelastic model in which each cell is modeled as a spring and dashpot. And we then made a series of such cells and imposed on them contractility according to the myosin amounts we had measured. And when we looked at the output, so here would be a profile of myosin. Here are resulting vectors between the cells of contractility. And and we let that run to equilibrium. And then we get different cell sizes. And indeed, the ones in the middle are small and the ones on the side are big. But this does not reflect the actual size relationships we see. So we thought, well, maybe the myosin measurements are sort of maybe a bit dodgy. Let's try all sorts of profiles. And we varied the width and the steepness and tried them all, modeled them all. None of them came out with the right answer, the answer that we saw live. Now, we had been using linear stress-strain relationships where, so materials that with a linear stress strain response means the stronger you pull, the bigger it gets. And conversely, the more you've already stretched it, the more power you have to put in to continue to stretch it. But there are other materials which, when you stretch them more, yield, so they become softer. Or there are others that get even stiffer, so it gets harder. So there are these different stress strain responses. And so we thought, well, let's do our whole parameter space again with these different responses. And it turns out, and here are 3 different examples, it turns out that for one of them, the elastoplastic one, so one that gets softer, where the tissue yields, actually comes up, at least one of the curves comes up exactly with what we observe. And when you superimpose these early curves that I showed you, the very best of the linear and the one of the many possible superelastic ones, that actually matches the data, which are shown in black here, much better. I was going to show you some nice modeling that we did with François Nedelec, who's now here in Cambridge, but I think I'll just go to the results. These are in silico models that are simulations based on active filaments, motors, and crosslinkers that correspond exactly to the properties we know for such molecules from biochemical measurements. And what Giulio, a former postdoc, did, he stuck a number of such cells, such models together, and then superimposed and imposed the same contractilities, namely myosin concentrations on them, and finds, again, tries a whole parameter space. And he too finds that for a certain set of parameters, we get the sizes as we see them live. So the amazing thing is that parameter space came up with the same conditions as the one from the viscoelastic model. So what we conclude from this is that there's a kind of tug of war. All these cells can constrict, but those that are in the middle and start a little bit earlier and a little bit stronger win and constrict, whereas those that are at the outside, although they can do it, are just too weak and too loose and expand. I have data that, so that's a hypothesis. It must mean that all cells have the capacity to do the same thing. We've shown that. It makes predictions. If you inhibit the strong cells in the middle, then the outer ones should be able to constrict now because they're no longer being pulled. And conversely, if you make the outer ones pull early, then maybe the middle ones will get stretched. And we used optogenetics to show this, but I will now let you get closer to lunch and not show you the data. Well, here's a bunch of stretching inner cells, and in the opposite experiment, there's a whole bunch of stretching inner cells. Well, sorry, previous one was constricting outer, and here is stretching inner. So we can impose other behaviors on them, not by modifying them themselves, but by affecting their neighbors. So this says that 2 cells which have identical genetic programs that determines their behavior actually talk to each other, where in such a way that an initial unequal recruitment of myosin leads to bias and constriction, which then further tears apart probably the myosin meshwork in the stretching cell, so that you get an mechanical instability and get a catastrophic slip and eventually a qualitative difference. These are the people who did it, very good lab members at EMBL Heidelberg. Some of the work was done using AI in collaboration with the Anna Kreshuk lab, and Giulio is a former postdoc who's now at NC State in the US. Thank you.
Thank you, Maria. That was an amazing talk. Do we have any urgent questions? Okay, one.
So when you do your model of myosin organization, do you postulate that myosin exists only as filaments of how many molecules, or do you postulate the possible existence of activated monomers?
It's active myosin. It's only activated. There's non-binding non-active myosin is silent in the model.
I know, but the mode of polymerization can affect.
We just model it as a, yes, indeed. No, we can do that. We can tweak any parameters. This is just as a dimer with, with a certain power stroke on the active filament. But we can, I mean, anything can be tweaked in that model.
Okay, I think we can continue the discussion later in the lunch with Maria. Thank you very much, Maria.